Hi, my name is Haley Zanga, and I will be talking about the promising outlook of mesenchymal stem cell therapy as a novel treatment for traumatic brain injuries. During this presentation, I will be talking about traumatic brain injuries, a brief overview of what they are, the pathophysiology that is behind them, and current treatment options that are available. I will then go in and discuss a new emerging treatment for traumatic brain injury patients, which is stem cell therapy. I will give you a little bit on background information and different sources you can use. I will then focus my presentation on one specific type of stem cell and look at its ability to possibly become a therapeutic treatment option for traumatic brain injuries. I will then look at preclinical studies that have been done using this mesenchymal stem cell in traumatic brain injury animal models. I will then talk about the conclusion and then data gaps and future research that is still needed. Traumatic brain injury is a major cause of death and disability worldwide. They are becoming more prevalent, affecting an estimated 1.5 million Americans annually. Traumatic brain injury is characterized as an alteration in brain function or other evidence of brain injury that results from an external mechanical force, possibly leading to permanent or temporary impairments. These impairments include deficits in social, cognitive, motor, and behavioral functioning. Traumatic brain injuries can be caused by many different things, such as direct impact, extreme acceleration or deacceleration, rotating forces, and outside forces that impact the head that causes the brain to rattle back and forth. Traumatic brain injury induces a complex pathophysiological cascade of cellular events, which consist of blood-brain barrier breakdown, widespread neuroinflammation, diffuse axonal injury, and subsequent neurodegeneration. Traumatic brain injuries can be classified into two groups: primary injury and secondary injury. The primary injury occurs at the moment of initial trauma and can include contusions, damage to blood vessels, axonal shearing in which the axons of the neurons are either stretched or torn, blood-brain barrier and meninges may be damaged, and neurons may die. The primary injury then leads to the secondary injury. The secondary injury is an indirect result of the primary injury. It occurs in the hours and days following the primary injury and plays a large role in brain damage and death that results from the traumatic brain injury. Following the primary injury caused by the trauma, an intense localization of inflammation occurs, worsening the damage already done and expanding the site of injury to include neighboring neurons. The primary injury causes a complex cascade of cellular events that are key characteristics of the secondary injury. These can include things like ischemia, impaired metabolism, free radical formation that results in apoptosis and necrosis of the neurons, and release of endogenous excitatory amino acids. To date, there are no pharmacological agents or effective treatments to improve neural structure and repair and functional recovery after traumatic brain injury. Current acute treatment of traumatic brain injury has been limited to prevent further swelling and pressure in the brain and optimizing cerebral perfusion to prevent further inflammation and cellular death, whereas chronic treatment focuses on cognitive, motor, and behavioral rehabilitation. The primary focus on treating traumatic brain injury patients is aimed at reducing the extent of the injury rather than repairing the damage. As of right now, there are little treatment options available for patients with traumatic brain injury. Thus, there is a need to develop further treatments. With the lack of current treatment options for traumatic brain injury patients, researchers have started to consider using stem cells as a therapeutic treatment option. Given their capacity to regenerate cells lost through disease or injury, stem cells offer a promising possibility to use as a treatment. Due to the limited capacity of the brain to repair and replace the missing tissue after injury, stem cell transplantation has now become a prospective treatment for traumatic brain injuries, since the transplanted cells have shown the potential to differentiate into region-specific cells and integrate into the host tissue to replace the lost and damaged tissue. Transplanted cells could also provide a type of nourishment support to the host tissue and help facilitate regeneration. Stem cell therapy has now become a hot topic for treating traumatic brain injuries since recent research has shown that stem cell therapy has positive therapeutic effects in patients with neurodegenerative diseases, such as Parkinson's, and was shown to be clinically safe when administered in patients with injuries such as myocardial infarction, stroke, and acute kidney injury. Using stem cells for traumatic brain injury treatment could potentially mean overcoming the damage done to the neurons in the brain. The goal is that the stem cells would work to repair or partially heal the connections between the nerves and cells within the brain. In the end, the goal is that this will help restore motor, cognitive, and behavioral functions that are often lost because of a traumatic brain injury. Over the past 2 decades, researchers have explored a wide array of cell sources for neurotransplantation. The most common ones include neural stem cells, embryonic stem cells, and mesenchymal stem cells. All three have been shown in previous research to be able to differentiate into a neural lineage, which is an important factor to consider when choosing a source. However, there are some challenges that come with these. An example of this is a challenge that comes with neural stem cell transplantation is the ability to deliver the cells to the specific area of interest, and these cells are also more difficult to isolate. Embryonic stem cells are great to use since they can proliferate without limit and become almost any cell type. However, there are ethical problems that surround these. Due to the obstacles of using adult neurostem cells and the ethical problems that surround embryonic stem cells, mesenchymal stem cells have now become a popular choice to use for stem cell therapy. Mesenchymal stem cells have become of interest to scientists since these cells are found in many and can give rise to many tissue-specific cell types. Researchers have also found many properties and abilities mesenchymal stem cells possess that may be helpful in treating traumatic brain injuries. I will go through and talk about each one. Like I stated earlier, it has been shown that mesenchymal stem cells can differentiate into neurocells. The ability of mesenchymal stem cells to differentiate into neurocells was first recognized by Sanchez-Romas and his colleagues. They showed that both mice and human mesenchymal stem cells had the ability to differentiate into neurons in glial-like cells under certain culture conditions. Furthermore, when stained with antibodies before implantation, this staining disappeared throughout the experiment, showing mesenchymal stem cells had differentiated into another lineage where mesenchymal stem cells acquired the phenotype of their host tissue. It was also found that mesenchymal stem cells are capable of secreting cytokines, chemokines, and growth factors. These factors secreted by mesenchymal stem cells, such as these neurotrophic factors, have important roles in creating favorable microenvironments for the proliferation of neural cells at the injury site. These soluble factors are capable of enhancing the survival of both stem and neuronal cells. These neurotrophic factors secreted by mesenchymal cells have been found to promote angiogenesis and neurogenesis in the injured brain, thus enhancing the multiplication of neuronal cells at the damaged site. Mesenchymal stem cells have also shown the ability to migrate to the site of traumatic brain injury. Many mechanisms have been postulated, yet no one knows exactly how this works. Mesenchymal stem cells have also been found to facilitate the injured tissue's own regenerative process. Mesenchymal stem cells implanted into mouse hippocampus was found to heighten the proliferation, migration, and differentiation of native neural stem cells. This mechanism is thought to be related to chemokines released by mesenchymal stem cells or indirectly via activation of the astrocytes. This was also shown when looking at lung tissue treated with mesenchymal stem cells. Researchers found a decreased expression of inflammation-associated cytokines, which facilitated the repair of the injured tissue. Over the last decade, researchers have been looking into strategies to enable access through the blood-brain barrier. Recent research has shown that mesenchymal stem cells have the ability to cross the blood-brain barrier. The ability to cross the blood-brain barrier is important for neurotherapeutic drugs. The mesenchymal stem cell's ability to cross the blood-brain barrier is unique and a promising feature supporting the usefulness of mesenchymal stem cells as a traumatic brain injury treatment. The use of mesenchymal stem cells have been brought to many scientists' attention in studies in regenerative medicine due to their easy access the abundance of sources, their potential to differentiate into many cell types, secretion of neurofactors, selectively migrate to damaged areas, and their potential to cross the blood-brain barrier. With the known advantages of the mesenchymal stem cells, more and more researchers have started looking into using mesenchymal stem cells in traumatic brain injury-induced animal models. A variety of studies have shown that stem cell therapy in mice and rats with a traumatic brain injury has had significant improvements in cognitive and motor functions. One study in particular, shown here, measured the effects of bone marrow stromal cells administered intravenously in rats with a traumatic brain injury. Rats were injected with mesenchymal stem cells labeled with BRDU into the tail vein 24 hours after the traumatic brain injury and examined throughout a 2-week period to evaluate neurological function using the NSS scores and the ROTAR test. As seen below, there was a significant reduction in the neurological deficits at day 7 and 14 compared to the controls. Figure 3. In Figure 4, it shows similar results where there was a significant improvement in motor function at day 14 after treatment compared to the control. These results suggested that IV administration of mesenchymal stem cells may be useful in treating patients with a traumatic brain injury. This is just one study showing that the use of mesenchymal stem cells in traumatic brain injury can improve motor and cognitive functions. Another study, shown here, also looked at the transplantation of mesenchymal stem cells into a rat model with an induced traumatic brain injury. Their study showed that some of the BRDU-labeled mesenchymal stem cells migrated into the parietal lobes of the injured brain, whereas the immunofluorescence microscopy of coronal sections showed no localization of BRDU-positive cells in the traumatic brain injury group, that's seen in Figure 5. This migration to the injured brain tissue was seen at 14 days after. In addition, double staining showed that some implanted mesenchymal stem cells expressed a neuronal marker and an astrocyte marker in the transplanted group. This is seen in Figure 6 and 7. They also showed that there was promoted functional recovery that was seen by the end of the 2nd week. Not only did they see migration and neuro-associated markers, but they also saw rat deficits that were improved significantly in the cell transplantation group compared with the traumatic brain injury group, the control, at 14 days post-injury. These findings are similar to the study that I discussed above. The advantages and properties that mesenchymal stem cells possess may have the capability of helping neural tissue damage that resulted from traumatic brain injury through stem cell therapy. Mesenchymal stem cells has shown us their prospective role in healing the damaged tissue by aiding in decreasing the inflammation, encouraging the regeneration of damaged nerves, and their tendency of homing near the injury site. Mesenchymal stem cells' ability to migrate across the blood-brain barrier would also overcome a major problem faced in treating traumatic brain injury, which is selective and target delivery to the injured brain tissue. Not only that, but numerous preclinical studies have shown reduced neurological and motor deficits in traumatic brain injury models. Stem cell therapy using mesenchymal stem cells has shown a promising outlook in the management of a traumatic brain injury, a condition that has once been resistant to previous intervention. A lot still remains unclear, and further research is needed to better understand and determine the best method to promote recovery of the damaged tissues. Longer-term studies, potential complications, routes of administration, and use of combination therapy are things that we need to look at. Looking at these things will help us explore the safety and efficiency of using these cells for a traumatic brain injury treatment. Thank you for watching.